Sure.
Oh, how do I mute my eyes? Unmute. Okay.
Cool. That must have been like for everybody.
Yep. Very fun.
Oh.
Okay. Hello, everyone. We're going to get started. Thanks for coming out to our second week of student presentations. It's nice that you're able to come here and support our students. We really enjoy doing this because we don't always get to interact, especially as we get up into older years. So it's nice to build community and see what each other is up to, and also have like a nice, safe space to celebrate both successes and failures, because they're all part of the learning process. So up first, we have Philippa Tanford. She hails from Indiana, went to undergrad at Duke, and is going to talk to us about plant diseases and honeysuckle. So let's welcome Philippa. Not talking about honeysuckle, just plant diseases. My bad, I got really excited about the honeysuckle. Moments while I get this situated. Maybe. Okay. Thanks, Anna, for the introduction, and thank you to the Living Earth Collaborative for having me. I'm going to talk today not about honeysuckle, unfortunately. Stay tuned for potentially some future talk about honeysuckle. But talking about some of my dissertation work examining diversity disease relationships in restored Missouri glades. So anthropogenic change is contributing to massive biodiversity loss. And this is, ongoing. We're in a species extinction crisis, habitat destruction crisis. And with these changes in biodiversity, we also can expect changes on ecosystem functions and services, any ecological process that any of us here might study, including disease systems. So with global diversity loss, we have can expect changes in community structure, community composition, and that can change the hosts and reservoirs that house and host diseases. So we can expect that these relationships between diseases and species diversity can result in either reductions in disease or amplification of disease. So if we plot species diversity and disease, we can expect that these changes could result in either one of these patterns. So let's look at an example. So biodiversity can mediate disease by a variety of mechanisms, but one that is easy to illustrate is what we would call encounter reduction. So on the left, we have a diverse range of hosts to Lyme disease. We're focused here on a focal organism, a human being, and a focal disease, Lyme. And these various hosts vary in their competence to host Lyme. So deer and possums are relatively less competent hosts to Lyme disease, while white-footed mice are much more competent hosts of Lyme disease. And in changes in this forest ecosystem, with decreased diversity, we end up with a much larger proportion of white-footed mice. And this leads to a higher disease risk with lower diversity for that human, because ticks that carry Lyme disease are essentially wasting bites on less competent hosts for Lyme. So conversely, we could expect that biodiversity can increase the prevalence of disease, which we would call amplification, with a wider diversity of host types, these green, blue, and yellow types of organisms on the right. We can expect instead that there are just more diseases available to that focal human. And therefore, potentially with a larger diversity, they might instead have a higher disease risk. So disease and diversity relationships have been studied in a variety of systems, not only animals and not only Lyme disease. And different studies have found different things, both amplification and dilution, or neither. And several meta-analyses have attempted to resolve these different results and found overall different arguments about whether, yes, we have a lot of dilution with increased biodiversity, or we have more amplification. And in Jason Rohr et al., in their meta-analysis, if you will, of these other meta-analyses, have suggested that one important factor that may help explain these discrepancies is the spatial scale at which these studies were conducted. So if you think about the smaller scale, where with these studies in this first panel have an average spatial scale of 1.5 kilometers squared, all the way up to more regional spatial scales here on the right, they find a trend in which at those small spatial scales, they see more dilution, where at the larger, more regional scales, they see more amplification, which can kind of make sense if you think back to that amplification diagram, where with larger, regional or up to continental, up to the globe assessment, you're capturing just more of the available reservoirs and hosts and diseases. So, but what about at even smaller scales? What if we're not thinking at the scale of kilometers, but at meters? Because those mechanisms of dilution, like thinking about the encounter reduction, those sorts of mechanisms should mostly operate at those very small scales. So in this study, published this year by Liu et al. In ecology, they studied plant communities and studied the effect of different plot sizes in whether they observed dilution. And you can see in the smallest scale up to the largest scale, they see a decreasing dilution effect. So at that smallest scale is when they saw that strongest effect. Okay, so what am I talking about today? So today I am asking several questions within this area. So how do biodiversity disease relationships scale in plant communities? How do edge effects, which I will define for you in a moment, influence these diversity disease relationships? And then, how do diversity damage relationships vary between different pathogen herbivore types? So the introduction was very pathogen focused, but herbivores as other enemies that cause damage to plants, we can consider in a similar conceptual manner. Okay, so my study sites are at Tyson Research Center, which many here are familiar with. It is WashU's field station for those who are not. We have St. Louis here in the river, and Tyson is within that white box. And then I have 3 sites along South Perimeter Road here for those familiar with Tyson. And each of these are large, round, they're circular sites that were made in an attempted glade restorations at Tyson glades, I'm not going to get too much into what a glade is because there's not a lot of time. I can talk about it afterwards. But for the purposes of this talk, these are mostly herbaceous plant communities. They're very hot and dry and surrounded by Tyson forest. These were cut out of Tyson forest about 10, 12 years ago. And it's maybe hard to see on the screen there, but those are the edges. And these are I sampled these plots, these white dots within each of them. So within each glade, I had 10 1-meter square quadrats in which I assessed plant species diversity and then pathogen and herbivore damage. So I took several metrics of each of these, including for plant species diversity. I measured species richness, relative abundance, and we will include metrics of phylogenetic distance. For today's talk, I'm only talking about species richness when I say diversity. And then for pathogen herbivore damage, I measured prevalence, which is just the presence or absence of that damage type. And I also, in a subset of the plots, I also measured severity, but today I'm just talking about prevalence. So these are the herbivory and disease types that I measured. I actually measured some others, but we didn't see enough of them in my data set to talk about them today. We have chewing and mining and sucking rasping, which are caused by different insects munching through plants. And then leaf spot disease and rust disease, which leaf spots can be caused by a variety of microbes. Rust is a fungus. Okay. And so at this, what are my hypotheses? At the scale of these 1-meter square plots, I hypothesized that the prevalence of disease and herbivore damage would decrease with plant species richness, indicating a dilution effect, since we are at these very small scales. And additionally, I hypothesized that the strength of this dilution would vary with the distance from the center of each glade. So you can imagine that with forest outside of these sites and more herbaceous, sunny plant communities inside these sites, that we have differences in microclimate factors, we have differences in plant species composition, and that these may, and then of course differences in things like moisture, which means that at this sort of outer region of these glades, we may have some sort of gradient zone or zones that are more affected by the factors in the surrounding Tyson forest. I did not a priori have a hypothesis about what this would look like. I could make a lot of stories about, a certain forest understory plants might be more likely to be here, but there's just a lot of different directions in which you could imagine that those might have an impact. But I thought that this being a different environment would potentially affect these diversity disease relationships. Okay, so starting with disease, we have here plots of leaf spot and rust. I did not find a significant relationship between species richness, and the presence of leaf spot or rust. The different colors of these points just represent each site, and each point is a plot. So on the x-axis, we have species richness. On the y-axis, we have the mean prevalence of that damage type. So I was assessing individual plants, and 0 is that symptom was absent, 1 is that symptom was there. So we have an average of an average of that damage type. So these are not significant. P-values are up here, but we do have a negative trend towards dilution, which will, yeah. So not sure yet what that means, since these are not significant results. For herbivory, we similarly do not have a significant relationship between species richness and herbivore damage, but the herbivory actually trends in the opposite direction. We have kind of an amplification trend, which is contrary to what I predicted. And I am still thinking about what might be happening here and would love to hear anyone's thoughts about it. Right, okay, so moving on to edge effects. So just to start out, there was no significant relationship between plant species richness and the distance from the center. So that means testing for sort of other patterns in the disease and herbivory diversity interactions wouldn't be confounded with, for instance, much more diverse plots at the edge versus less diverse plots in the middle. Yeah, and there is also similarly not a significant relationship between the pathogen herbivory damage and the distance from center. In similarly non-significant results, the richness damage relationship doesn't show a significant edge. So these plots, each of the points again are 1 meter by 1 meter plots, and the color indicates the distance from the center of the glade. So these darker colors at the bottom of the scale are closer to the center, and the lighter blue are closer to the edge. And I do not have these plots included, but we did test for a significant interaction as well of distance and the damage richness relationship, and did not find a significant interaction there. 2 minutes. Cool. Okay, so why do we not see significant disease dilution as I predicted we might? So one possibility is that average prevalence could be the wrong disease metric to see dilution. Severity is, there's a bit more range in severity. We had some plants, for instance, that almost always had chewing damage. And so then the significant, pattern could emerge in how much of each of those plants was chewed or, and I do have severity data for a subset of these plots, so I will be looking at severity. There could also be some site-specific differences in host species or dilution amplification mechanisms. Each site had a similar overall plant species richness, but some of the constituent species vary, and just anecdotally in ways I did not test, they looked somewhat different. And we did do site-specific tests, but those were not significant. Excuse me. But on some of the damage metrics, they do trend in different directions, which could indicate site-specific differences. Yeah, I'm not sure yet what I think about the amplification trend with herbivory. We will maybe need to first see if it's significant when we look at severity, but that's something to still think about. And perhaps just the edge environment of these sites is not distinct enough to see a difference in these patterns. Yeah, so I will be analyzing these with other metrics of diversity, thinking about how these patterns scale spatially with different sort of neighborhoods of plots, and also be collaborating. This protocol is part of a larger collaboration based in Europe. It's called BugNet. You can look at, I don't have the website's in acknowledgements, bugnet.org. And they designed the protocol that I used. And thank you very much. We have time for one quick question. All right.
I'm sure you noticed that of your what was it, 3 herbivores, the
Mhm.
relationship was all positive.
Yes,
With this was for the 2 pathogens, it was negative.
yes.
Would be interesting to see what combining those 2 separately would show statistically. I think it's hard to do because you would be generating a hypothesis from your data, and perhaps not a fair thing to do, but it would have been one of the natural hypotheses to test, perhaps.
Yeah, that's a great idea. Thanks, Dave.
All right, great. Let's give Philippa another thanks. Okay. I'm gonna try to introduce our next speaker and switch the files at the same time. So up next, we have James Ojascastro, who went to undergrad at Colby College. Did I get that right? Sweet. And is in the lab of Robbie Hart and will be talking to us today about paper.
Good afternoon. Yeah, thanks Anna for emceeing and for LEC for hosting this. I'm going to talk to you today about paper. As academics, we should be quite familiar with papers. I mean, most of our time is spent reading them and then after a few years writing them. Who do we get to blame for this academic tradition? Well, about 2 millennia ago, this official from the Han Dynasty court in China is said to have discovered that when he beat up some paper fiber, some vegetable fibers, you can do this either physically with a mallet or using some alkali to macerate them, and you get this pulp. And so if you distribute this pulp on a screen and then take out the water, it'll dry and those fibers will felt together using hydrogen bonding, and you can make a laminar sheet. Well, it turns out that there is some evidence that paper was made before Cailun. This is the earliest evidence. I believe it was maybe about 200 years before his lifetime. But actually, maceration and felting of vegetable fibers occurred following a tradition that dates back perhaps 8,000 years. So this is Tapa. There is an ethnographer from Germany who's long since passed, but he asserted that paper came about because there was this transition between felting vegetable fibers for clothing, and then people would decorate them. And they discovered, well, you can use that to write your history as well. So given that we have some 8,000 years of history and knowledge here, some of it is maybe pulp fact, and various people have documented that, and some of it maybe is pulp fiction. So there's a lot of clarification that needs to be done. There's some unanswered questions. One of them, let's take all plants, the whole plant kingdom. All plants, if you look at their cell walls, they're made out of cellulose, and these celluloses can be put together something like Legos. This bonsai tree is made up of these tiny components in the same way even giant sequoias are made up of these tiny little parts that fit together. It's all cellulose fibers. But what's interesting is that given that all plants are made of cellulose primarily, and paper is also predominantly made out of cellulose, do we see traditions of processing these fibers indiscriminately, irrespective of species? The other question is, are all those fibers within the same plant also equivalent? And the interesting thing is no, because people who have processed these fibers by hand, most of them are pretty flexible in other tissues, whether it's from the phloem or bast, from the leaf, Grasses have kind of a complicated physiology within their stem with the phloem and xylem mixed together. And then fruits, things like cotton, you can see these cellulosic fibers in the fruit. Wood is not readily accessible to many artisans because it's rich in lignin, and that lignin makes it very hard to process by hand. It turns out there's some proof of concept here. So I'm gonna share a binder of some paper examples just to prove to you that these cellulosic fibers from different plants can be processed into paper. Here's some examples from a publication about 20 years ago, some from ponderosa pine, cattail. Some examples that I made that you'll see in this notebook, some from pawpaw and some from wisteria. But you can see that the personalities, the qualities of these papers vary considerably. Well, why is that? If we look at the traditions from several countries, things that have been practiced for centuries, they have converged on using very specific species to process into paper, and that have been practiced in this way for centuries. And so they've picked maybe 1 to 3 different plants in each of these countries. And this is true around the world. It's true also in Mexico. And so these traditions, over time, they've found ways to transform the fibers of very specific parts of very specific plants into highly refined papers that can last for centuries. And if you ask me, this is some serious gourmet paper. So here's kind of our problem. We have 300,000 or 400,000 species of plants all around the world. But if you look globally, only maybe about 50 to 100 species have been processed into paper by hand. And so why is that? Well, if you look at the qualities of the plants that are used for these traditions, there's maybe 5 different qualities that I like to emphasize. But for the sake of this talk, I can only focus on 1. So we can actually measure these qualities or functional traits of those fibers. And there's 4 key measurements that you can make. So these fibers are made something like drinking straws. You can measure the length, the diameter of the hole in the middle, the lumen, and then the thickness of the cell wall. And from this, we have kind of a 4-dimensional space. So keep that in the back of your head, right? What we can do is also set up a comparison. So we can look at fibers from plants with a tradition in hand paper making. So these I'm going to call paper plants. And then you can also look at plants with no evidence of this kind of tradition. So something like burning bush, for example, right? And just to emphasize that all of these fibers are in theory or in practice useful, right? We have examples in that binder I'm passing around. We have Mitsumata paper, which has been made in Japan for 400 years, and then some experimental paper from burning bush. We can look at those fibers under the microscope. So I've been doing that in the basement of McDonald. And you can see that the physiology of these fibers under the scope does look by eye different. I know it's a little hard to see, but you can actually click in and make these little lines using that software to show how long that fiber is. And so it's a little difficult to see these different colors, but it looks like the shape and size of these fibers between these species is a little bit different. Okay, so I've been measuring 4 different metrics related to these fibers. And because it's 4 dimensions, we have to find a way to visualize that. And so one way you can do that is through statistical ordination. So what I chose to do is I made a plot with non-metric multidimensional scaling. And so for those who are tuning in who are not familiar with this, the idea is that you can show a scatter plot with 2 axes. And the idea is that points that are sampled in this statistical technique, those that are closer together spatially tend to have very similar characteristics. Those that are far away tend to have very dissimilar characteristics. And so this is what that plot looks like. And so I've color coded this for you to show the differences between paper plants shown as PP and teal, and then non-paper plants shown in red. And spatially, we see that there's a little bit of a difference in exactly where paper plant species, judging by their traits of their fibers, occupy in this space relative to non-paper plants. And so the question is, what explains this particular spread of these points? And it turns out the category, whether or not a plant is a paper plant or not, is statistically significant in explaining the differences in physiologies of different fiber plants. But it's not just that, it's also what tissue is used of that plant. And also we know that evolutionarily speaking, plants with similar traits oftentimes are also evolutionarily related. And so we can also see that the identity of the family that these fibers originate from also significantly explain this spread of points. What's interesting is that we have this kind of interface where paper plants and non-paper plants meet in this ordination space. And I want to investigate that a little bit more. So in that binder, there's some paper I made from cultivated fig, and I also measured it under the scope. The fig is shown in red, and you can see those 4 metrics shown in microns in that table up there. And flax and paper mulberry and Mitsumata are all used in traditional paper making. And you can see that cultivated fig seems to have fibers that are comparable of the metrics that are shown for fibers used in paper making. And so why is it not a paper plant? Well, maybe there's some cultural trends involved. And so I posit that perhaps priority effects are playing a role. So for example, if you pick a plant and you're going to designate using that in a particular way, traditions kind of have this inertia where it's difficult to displace a plant that is currently being used with something new. On the other hand, there are people who really like to experiment. And so that kind of push and pull, I believe, explains why some millennium has passed in Japan where people use only 2 species for traditional paper. It's not until about 1600 that Mitsumata is adopted. The other reason is that there might be a competing use. So if you're using a plant in a certain way, figs are very tasty, in my opinion. You might not want to use that plant for paper. You might want to use something else that is inedible. Okay, so what can we conclude from this? Despite all plants being rich in cellulose fibers, we can see from this ordination that our ancestors were extremely discerning in tailoring plants with certain traits for paper and clothing. However, explaining how and why we use these plants or don't use these plants, that can be pretty complicated. So far, what I've shown you is that we see that there's a selectivity among artisans in using plants with particular characteristics for a particular purpose. What I haven't told you is, well, why? And so what we need to do is actually test the paper that comes from all these different species and figure out, well, what co-varies with these particular fiber characteristics. And we can test that using some antiquated machines that measure tearing, double fold. So you can put a little piece of paper under stress and then fold it back and forth and see how well those fibers resist that stress. You can use a tensile strength meter to test how much force does it take to pull apart those fibers, literally rip it apart. So that's something I'm looking forward to doing this spring with a whole rainbow of different fibers. I want to thank a lot of people who have given me money and also I've worked with, including my advisor, Dr. Robbie Hart. So with that, I can take questions. Thank you.
Really nice talk, James. Thanks. 6,000 years is a long time. Is there any evidence for domestication of species? for plant making compared to their wild ancestors?
Cotton's a really good example. People have hybridized the different species from West Asia, from India, and from the New World to try and get not only good fibers for making paper, but also for different textiles.
I have one question. You mentioned at the very beginning there were a couple different paper production techniques like maceration and alkali. Do those processing techniques have any effect on fiber characteristics?
Yeah, so that's a good question. There's traditions in the West, European traditions of processing paper, and then there's traditions in the East. And they use slightly different kinds of plants. So in the European tradition, they use flax and hemp and things like that, things with very exceedingly long fibers. And so what you actually have to do to make it not clump and make uniform sheets is that you need a technique that actually cuts those fibers into shorter lengths. And in doing that, you'll get smaller pieces that more closely resemble the fibers that you see in the bark of woody species that are used in East Asia. To contrast that, there is also a machine called a naginata in Japan that instead of actually chopping those fibers, will shear the fibers away from each other without sacrificing their inherent length. One thing I do want to note when I measure these fibers, fortunately, the maceration is very gentle, and so I'm still able to look at a microscope and see, well, is this fiber still intact? And you can see that many of the fibers taper into like knobs or like actual points, whereas those that are cut visibly have a cut.
Okay, let's give James another thank you. Okay, up next, we have Sean McHugh. He is a second year in Michael Landis's lab, went to undergrad at Cornell and has his master's from Virginia Tech, right? Super. So here to talk to us about some biogeography, here's Sean.
Great, thanks. Yeah, so how's this going? All right. Okay. All right. Hi, y'all. Thanks for coming. My name's Sean, as we said, and I'm largely interested in developing computational methods for modeling niche evolution over macroevolutionary timescales. And in this case, I want to talk to y'all a little bit about a model I developed for estimating realized and fundamental biome shifts. Now, to start off, let's just discuss a little bit what is a biome. Now, biomes are really complex and it's really hard to nail it down to a single definition often. However, in our case, we're really focusing on the definition that there are major habitat types with consistent ecological and climatic features as a large spatial assemblage. And we have a few instances of them right here, like tropical rainforest, temperate forest, and desert, each of which having pretty consistent climatic and conditions and ecology. So oftentimes when we're modeling these over macroevolutionary timescales, we're often interested in modeling the discrete shifts between different biome affinities, and in this case, we're showing an instance with a present day observation of a species in a temperate forest. And working back through time with a normal model, we'd be detecting shifts along the phylogenetic branches we're showing here, and we're working back in time. So in this instance, lost a tropical affinity, and it used to be a generalist ancestor. Go even farther back in time, and it has been actually just a tropical ancestor that gained a temperate affinity and lost a tropical affinity. This is a simple example over just a single branch and show what it looks like over a very small phylogeny. And we just show a stochastic map here often would be where we're showing a series of events of gains and losses of different biomes. And most notably at speciation, we often try to account for asymmetrical inheritance of biomes, where one daughter lineage may only get one biome affinity, another daughter lineage will get the other. And these are really useful tools for detecting a lot of different macroevolutionary patterns and not only reconstructing what the ancestral states for biome affinities for species, but also the rates of transitions between given biomes. However, one challenge with a lot of this is we're going off of what we observe here. This is what dictates most of what we understand about biome affinities. We see where they are, we identify those as the biome affinities, and then we go from there reconstructing back. But we have a lot of instances where we see species that thrive outside of their native biome. I'm going to go with some low-hanging fruit here to start with the monk parakeet, where it's temperate grassland, and you find it in an urban setting. Now, urban isn't a traditional biome, of course, but it really just shows that it's able to survive well outside of its natural range into other environments. And there's a lot of examples of species inhabiting novel biomes that are natural outside of their range. And showing a few examples here. Yeah, the ants aren't great. So just to give an example of kind of why this matters, because I'm going to get into this terminology of realized versus fundamental niche here, and I'll try to be gentle here. So here we have an example of a species with its realized range over a temperate biome. We'll just consider this a simple biome space. So its realized range. It is found in temperate biomes. Simple as that. And then we see an instance where somehow it shifts over and accesses a tropical biome as well, and it occurs in both. Now, how did it get there? Now, if you just look at the realized niche, probably adapted and evolved there, and that's totally fair. That could happen. Consider a fundamental niche. Its full range of abiotic conditions that it could survive in is well within the temperate range. In this instance, for it to evolve, it would be expanding its fundamental niche, gaining new physiological, ecological traits that allows it to thrive within a tropical biome. However, that's not the only way things can access a new biome. Let's run this back another time, but with the same realized niche shifts, but now the fundamental niche was always covering both biomes. And it just moved to access this new biome, or the environment had shifted underneath it, and it became a new biome, but it was still within its range of conditions it could thrive in. And this is often a big question of interest for us. When do species move versus when they evolve? And that's something that I think we could all kind of get behind as being a little interesting, hopefully. But yeah, so to go back to that brief example over a phylogenetic branch before, right? So simple temperate gain, tropical loss. Well, this is really just the realized affinities that we're tracking here. Under our proposed new Bayesian model, which this is a work in progress name, but RealFunBS, realized and fundamental biome shifts. I can tell you this is not the worst name we could come up with. There was far worse. But anyways, so we're going to work through a few steps here, going from past to present day here, just to show what the evolutionary process would look like. And you start with, in this case, a temperate gain in the fundamental niche. It evolves to be able to thrive, able to persist in temperate biomes, but doesn't access them. Then it gains the realized affinity and is able to actually access that environment. But then lost the tropical. It can still survive in them, but something changed. Biotic interactions, its dispersal axis, the environment shifted out from under it, no longer in the tropics. And then it eventually loses that fundamental loss. Now, this is a very step-by-step process, which might not always be realistic here. Sometimes you could totally go from having no affinity for something to having the affinity and being there, gaining the realized and fundamental. And that's cool. We account for that as well. And to put this over the map that we were looking at before, this is what's just, again, it's now really just realized affinities, where we have a gain of realized temperate loss of realized temperate and tropical, the asymmetrical inheritance. But let's try to fit in what what it would look like when considering fundamental affinities here. So we have here at the ancestor had a fundamental temperate, perhaps, had at speciation, it was an asymmetrical inheritance of the range, but they both fully inherited the fundamental biome affinities, which is an instance where allopatric speciation or something else, versus other instances where you could have the full split and have it where each one only has the fundamental affinity for one of the biomes. There's a lot of ways to deal with speciation. I'm only glossing over it. Happy to go into more details if anyone's interested. But yeah, until we get to the end, you lose a fundamental biome for the tropics, and then you have a distribution that looks a little different from what you see just looking at the realized affinities. And what that's the hope with our model, is be able to account for these fundamental affinities both back in time and try to estimate in the present as well. Granted, data is hard, but we have fully implemented this model so far in a Bayesian framework. I'm not going to go through the slog of simulations to show how it works, but if anyone's interested, happy to go through it. Very happy to go through it. But so we're going to just show here how we're going to apply RealFunBS to viburnum, and a clade with a really dynamic history that has occurred across quite a few different biomes, everything from the tropical rainforest to cloud forest that's like warm temperate, all the way to cold temperate forest. And we'll get to that. And across it, you see distinct changes in leaf morphology that shows a really adaptive shifts towards these different climates. But there's a lot of shifts over the clade. And so to the extent where there might be some realized, some fundamental affinities that might be worth tracking. Is it really where it's shifting adaptively each time when it's shifting between daughter species, or is it really potentially just moving between biomes? And so here's an estimate. This is a full ancestral state reconstruction. I'm not going through this whole thing. I don't think I can even see the dots from here. So I've blocked off this little subclade here just to show proof of concept, how it works. Now to line up here, the colors are the biomes, the fundamental affinities, and anything with a dark dot in the center that has a realized affinity. And the darker the color, the higher the probability that it is, the less transparent, the higher the probability that that's there. This is from sampling over an MCMC, many ancestral state reconstructions. So let's show this little clade here. Sure. So with RealFunBS, we have some really interesting patterns here where what we're seeing a lot at the tips is really supporting single realized affinities with some weak support for tropical affinities back in time. But I really want to focus here on the fundamental cold temperate affinities where you don't really see the dark dot anymore. It's not really showing much support for it physically occupying those biomes, but it's still showing support or a reasonable probability that there's fundamental affinities there. So in a clade that's wobbling back and forth a lot between temperate and warm, cold temperate and warm temperate, cloud forest, it's not always losing the realized and fundamental. It's just sometimes losing the realized affinity. And I wanted to compare, like, well, this is great. This is a model. What's it look like with other models, the way we usually try to approach this? And to compare side by side, I have the standard model we use to approach this, a DEC model, dispersal extinction cladogenesis, which again is only accounting for those realized affinities that I showed before with those little diagrams and demos. So how does that look? Well, here we see, and again, we have the dark dots in the colors, but they're both linked together. They're both the same transparency. The fundamental and realized are linked, and they're not being modeled as two separate processes and patterns here. And so in this instance, we're seeing a lot fewer colder temperate affinities in general. A totally different, a pretty different pattern in some ways. And it's just in general showing a lot less prevalence of cold temperate affinities, and a lot of those ancestors that RealFunBS is saying might be there, but further testing will show if that is BS or not. I had to say it, I'm sorry. But anyways, not only are we able to show cool patterns of ancestral state reconstructions, we're able to show also by estimating the rates of change between different biomes. We're showing we're just getting different processes here. So I'll try to work through the legend here for you. I'm just showing the gains and losses of realized biome affinities, simply because that's what we can compare between the standard models and our own. If anyone's interested in the parameter estimates for the fundamental affinities and such, happy to go into that. But here just showing for losses, these are the posterior distributions for those parameter estimates. So rates are the x-axis, density of the posterior distribution is the y. We're showing the distribution, so showing a lot of support for the same area of loss rates. But for realized affinities, the red is the DEC, the standard way of approaching things. The green is the same realized gain rates, so they're much higher. And then we also plotted blue, which is the rate of gaining both a realized and a fundamental affinity at the same time in one big jump. And we show that it is a rare event, but it is still present. So I wanted to show this figure here for a couple of reasons. One, shows processes are acting differently. Our RealFunBS is able to get a little bit more of the details and show different processes explaining these patterns, whether it is a single shift or a double shift where you evolve and move, or just evolve or just a move. It allows you to help tease apart these details. But we, of course, have a lot more to do to get this in proper working order. This is just a trial example. We have much more we want to do to check the model adequacy of our results, the adequacy of our model compared to DEC models and such. And also we have other hypotheses we're really interested in, such as, do occupied biomes impact rates? Right now, we've just implemented this in a simple scenario where rates are different between gaining realized or fundamental biome affinities, but the rates between going to different biomes isn't different. So that's a real area of interest, both for seeing, do the rates differ when accessing different biomes? And do the rates differ depending on the biomes that you already currently occupy? And if that does matter, what matters? Where you actually occupy versus where you actually have a fundamental affinity for. In addition, the low-hanging fruit with the urban system, those aren't natural biomes that evolved over phylogeny, but the traits that allowed the species to be pre-adapted did. And so perhaps it's worthwhile to try to map and model the fundamental affinities to some of the anthropogenic biomes that we see today and work backward from there and see how many species are pre-adapted, predict species that might be pre-adapted to urban biomes, and as well as just see what other covariates are tied to that, either traits or other components of historical contingency. So well, that's my spiel. I'd like to acknowledge the Landis Lab in its entirety. Everyone's been a huge help. And I'd like to take this moment for questions. Thank you. Hi, Robbie.
Hi, great talk. Really fun to hear that. I was wondering, how do you parameterize even for extant species, the fundamental biome?
That's a good question. So when we didn't have, so we tried to, where information was available, rule out. So we didn't just treat it as the realized affinity was the only thing that could be at the tip. So we allowed uncertainty, so it assigned at worst equal probability for all potential states with that realized affinity and other fundamental affinities. It wouldn't allow one which had a realized affinity for, say, temperate and tropical if we only observe it in temperate, but it would allow for, say, like the fundamental affinities for those. So we weren't biasing it towards the realized. And what we did try to do when we had instances where, like in viburnum, it was cold temperate, warm temperate, tropical. If you're tropical, you're not likely going to have a fundamental affinity for cold temperate, but not warm temperate, right? So we tried to rule out those sort of states where you didn't have the fundamental affinity for the middle ground, but you did for the other two. Does that help clarify? A big thing is, and I'm hoping to talk to the plant people here, is we are trying to figure out ways of ruling out when it is a realized only state. And if we have information on fundamental affinities, being able to rule out the instances where it is just one realized affinity and no fundamental affinities, that'd help our model quite a lot. That'd be interesting. But yeah, anyways, shameless plug for stuff there. Yeah. Oh, sorry. I'm not the one who's doing that.
Does your model take into account the distance like of a current, like of species like range to the different potential biome types?
That's a great question. At this current time, no, but that is something we're very interested in. Jointly inferring these biome shift histories with biogeography would be really cool. There are some details at that, but we are very much interested in that. That'd be a great step forward. Thanks.
Any last questions for Sean? No? All right, let's give another round of applause for all the speakers. And if you'd like to ask anyone any more questions, you can join us at the reception across the hall. Thanks. Not real. That was so great.